two components of the genetic pathway in C. elegans encode members of the BCL2 superfamily, AGO1 and Z9. What do Z4 and Z3 encode? The Z4 gene was cloned even before the Z9 gene, and at the time it turned out to encode a protein of 546 amino acids with no similarity to any other protein in the databases, and also with no known motifs. Junyin Yang in Bob Horwitz's lab, who had cloned Z4, teamed up with colleagues in the Horwitz lab and also cloned the Z3 gene a couple of years later. And with the cloning of Z3, she hit the jackpot. The Z3 gene turned out to encode a protein that had significant similarity to a protein in the database, and this protein had already been shown to have a specific enzymatic function. This protein was mammalian ICE, which stands for interleukin-1 beta converting enzyme. And ICE had already been shown to be a cysteine protease. Proteases are enzymes that can cleave peptide bonds between amino acids and therefore proteins. Cysteine proteases are proteases that have in their active site the site that's essential for peptide bond cleavage, a cysteine. Overall, the nematode Z3 protein shares 29% amino acid identity with human ICE. What was known about ICE? ICE had been shown to cleave the inactive precursor of interleukin-1 beta, a signaling molecule. Furthermore, it had been shown that ICE cleaved interleukin-1 beta between an aspartic acid residue at position 116 of the interleukin-1 beta sequence and an alanine residue at position 117. In addition, it was known that ICE is synthesized as a proenzyme or zymogen, which needs to be activated by proteolytic cleavage. This cleavage is done by mature active ICE protease, and the cleavage occurs at 4 aspartic acid residues within the ICE protein. The mature ICE protease is composed of 2 subunits, a P10 and a P20 subunit. Interestingly, 2 of the 4 cleavage sites within the ICE protein sequence were conserved within the Z3 protein sequence, which suggested that the Z3 protein might be activated through proteolytic cleavage as well. Indeed, Ding Xu in the Horwitz lab subsequently showed that the Z3 protein indeed is a cysteine protease that is synthesized as an inactive zymogen and that becomes activated by proteolytic cleavage at 3 aspartic acid residues. Cleavage at these sites generates a P17 subunit and a P15 or P13 subunit, which together form the fully active protease. Just like for ICE, this proteolytic cleavage is done by the Z3 protease itself. This was great. Z3, which is required to irreversibly commit cells to the apoptotic fate, encodes a protease, an enzyme that can irreversibly cleave other proteins. Furthermore, this protein is made as an inactive zymogen that needs to be matured in order to become fully active. Could it be that it is present as an inactive zymogen in all cells, and that it is activated specifically in those cells that are programmed to die? Could it be that its activation represents the point of no return? What are the substrates of these proteases? What proteins do they cleave? When Z3 was cloned, there was another important question that needed to be answered. And that question was whether ICE or ICE-like proteases also could commit cells to the apoptotic fate and induce apoptosis in mammalian cells. Basically, is the role in apoptosis of cysteine proteases such as C. elegans Z3 conserved? So that's what Junyin Yang decided to work on next once she had her own lab at Harvard. And the way she and her co-workers addressed this important question was by generating a number of different ICE and Z3 constructs and by expressing those under the control of a strong promoter, an actin promoter, in rat fibroblasts grown in culture. The morphology of cells expressing the various constructs was then analyzed using light microscopy to determine whether they acquired a morphology typical of cells undergoing apoptosis. And what they found was very significant. First, they found that yes, the expression of ICE in these cells could induce apoptosis. And required for this were both subunits, the P10 and the P20 subunit, as well as the active site cysteine. ICE can induce apoptosis in mammals, and its ability to do so is dependent on its protease activity. The role of cysteine proteases in apoptosis is therefore conserved from worms to human. Second, they found that the expression of C. elegans Z3 in these cells also induced apoptosis. And again, the active cysteine of Z3 was required for its ability to do so. The protease activity of Z3 is therefore also critical for its function in apoptosis. The fact that C. elegans Z3 could induce apoptosis in mammalian cells furthermore suggested that ICE and Z3 might have similar substrates and that the cleavage of these substrates might be important for a cell's commitment to the apoptotic fate. Finally, apart from expressing these different ICE and Z3 constructs in normal rat fibroblasts, they also expressed them in rat fibroblasts in which BCL2 was overexpressed. BCL2 is homologous to C. elegans Z9, and in the C. elegans pathway, Z9 acts upstream of Z3. If Z9 is active, Z4 should be inactive, and so should be Z3. Based on this, one might speculate that in rat fibroblasts in which BCL2 is overexpressed, the expression of either ICE or Z3 might not be as effective in inducing apoptosis. And that's what they found. The expression of BCL2 blocked ICE-induced apoptosis in these cells almost completely. In the case of Z3, the effect was much weaker, but some suppression was observed. This was an important finding. While quite a bit was known about the BCL2 superfamily at the time, that there are pro- and anti-apoptotic members, that there are sensors, guardians, and effectors, that they localize to mitochondria, and that they can bind each other. However, what was still completely unclear was what all this was good for, what the BCL2 family was actually regulating. The result in rat fibroblasts suggested that the BCL2 family members were involved in controlling the activities of ICE-like and Z3-like cysteine proteases. How they did this was still completely unclear. The cloning of C. elegans Z3 and the work with mammalian ICE that demonstrated that the function of cysteine proteases in apoptosis was conserved were published back-to-back in the journal Cell in 1994. These two papers were seminal, and they moved the apoptosis field a big step forward. C. elegans Z3 and mammalian ICE represent the first members of a family of cysteine proteases now referred to as caspases, which stands for cysteine-dependent aspartate-directed proteases. In C. elegans, there is Z3, and there are 3 additional Z3-like caspases, whose functions are currently being analyzed. However, they do not have a function that is redundant with that of Z3. As of 2011, there were 10 caspases in humans, and at least 7 of them have a role in apoptosis.